Okay, it's 2:03, so let's get started. So feel free to ask a lot of questions in the chat. We have a little bit of a small, more intimate group because I turned off registration at 20 people. So that way you guys can ask more questions and I'll actually be able to stop and help. So today's lesson is about gene expression. If you guys wouldn't mind private messaging me or messaging in the chat, what is like your level of experience with biology? Like, are you in middle school? Did you take biology class? Did you take AP biology? That will help me adjust the difficulty level. Okay. So first, I'm going to introduce the concept of. Okay, thank you to those who have given their feedback. So first, I'm going to do a very general review of the central dogma of biology, like DNA, RNA, all that stuff, to get everyone caught up to the same level. So in each cell, we have our DNA, which you probably have heard of at least once. And the DNA has the instructions for making everything in your body, such as like your eyes, your skin, enzymes, proteins, etc. So it's very important. And DNA is composed of 4 nucleotides, or you can also think of them as letters, that encode various amino acids, which I'll get into later. So there's 4 base pairs, A, T, C, and G, adenine, thymine, cytosine, and guanine. And basically, when you put them together in various combinations, they code for different amino acids, which are the building blocks of proteins. Each 3 letters in a row represents, well, encodes an amino acid. So what that means is later in the process of gene expression, if the ribosome, which I'll also explain, sees an ATC, it's going to put in the corresponding amino acid to the end of the protein. So you can think of an amino acid as a building block of a protein, and a protein is just multiple building blocks attached. So you could look at this Lego picture and think of each individual brick as an amino acid, and the whole thing comes together to form a protein. So what exactly is a protein? So proteins are small molecules in our body that do almost everything. For example, they attack bacteria, break down viral DNA, break down the food you eat, and even help you move. That's why athletes eat a lot of protein to get stronger, because those proteins can be broken down and then reincorporated into muscle fibers. So here in the picture, you can see some more ways that proteins are found in your body, such as keratin, which is kind of like a very tough fiber. You got phylensin and rhodopsin and factinin, and those are in your eyeball and they help you like transmit light through your eyeball. And there's also like neurofilaments in your nervous system, your hair, muscle, blood vessels, all that is composed of protein. So how to make proteins? We can think of DNA like a recipe book. It has instructions for making a lot of different proteins, thousands in fact. So you start off from your DNA and there's 1 in each cell. Well, technically 2 copies in each cell. You transcribe it into RNA, and the RNA is kind of like making a photocopy of a specific recipe in the cookbook. So for example, if you wanted to make a gene, each mRNA would have the instructions to make a gene, and the gene would be like a recipe in this analogy. Then the chef in this scenario is the ribosome, which takes in the RNA, which is the photocopy of the recipe, and then it makes it according to the instructions. The amino acids or building blocks are the ingredients used to make proteins. I'm not going to explain tRNA or transfer RNA today because it's a little bit more complicated to explain. So transcription is just a fancy word for copying. If you've ever heard like your teacher say like, oh, please transcribe this text. So script, you know, the word script inside, like writing as writing script. So let's say you have a recipe book and your friend is a chef and they want to use a recipe inside it, but your recipe book is very special and expensive and you don't want to give it away. What you can do is you can take a picture or photocopy one of the recipes inside the book and then give it to your friend. And then they can use that copy. So it's similar for DNA. So an RNA polymerase is kind of like a photocopier, and it comes along and it makes a copy of a specific gene inside the DNA, which is called messenger RNA. So in this scenario, messenger RNA would be the picture of the recipe that you gave your friend instead of giving them the original recipe book, aka DNA. RNA polymerase is kind of like a train that chugs along one strand of the DNA. So DNA comes in a double helix form, which you may have, you probably know about. So that's actually the name, where the name of this organization comes from, Helix Initiative. So anyway, RNA polymerase comes along, it picks one strand of the DNA and they get separated and it just chugs along, it attaches to one strand and it chugs along. And for every new base pair it kind of walks or runs over, it's going to add the complementary base pair to it. So the adenine, if it sees an adenine base pair, it's going to add a uracil. If it sees a cytosine, it's going to add a guanine. So the one thing to note about RNA is that instead of T for thymine, that nucleotide, it has a U for uracil. So how do we turn a messenger RNA into a protein? Your friend, the chef from the previous analogy, is the equivalent of a ribosome in the cell. Ribosomes take in messenger RNA, like the messenger RNA gets fed through the ribosome, and then the ribosome makes the protein one by one according to the nucleotides that it sees. The ingredients of the recipe are the amino acids of the protein. So just a quick review, we start off with DNA. The DNA gets transcribed into RNA for a specific gene, and then the ribosome translates the RNA into a protein by adding on an amino acid one by one. Gene expression is essentially how many proteins are made from the instructions of a specific gene. High gene expression means that there's many proteins made, and low gene expression means that there's few proteins. So one example of this would be, let's say you have a gene that provides instructions for, how about height? Let's just pretend there's a protein that makes you taller. So high gene expression would mean that the gene that codes for the height growth, a lot of the protein are made. Proteins are really important, as we've seen, but that means that when things go wrong and you get sick or you get a disease, they're usually or oftentimes behind it, or at least a factor in it. For example, cancer is when, great question. So that is a really good idea, but so multiple copies is actually a separate thing. It's called like multi-copy variants. So the thing about gene expression is that the gene itself in the DNA is not being changed. So there's just the one copy of the gene, but what is changing is how many RNA is being made, because we only have one copy of the gene in our cell, but how you can make more of the protein, which is this thing that actually is the end product, it actually does something, is by making more of the RNA. So the more messenger RNA you make based off of the DNA, the more I guess, the more copies you make, the more photocopies you make of the recipe, the more chefs can use the recipe to make more food, right? But you're not like having multiple copies of the DNA itself. Yeah, but the multiple copy thing is also a thing in genetics, just not in this case. One interesting kind of thing is that women have two X chromosomes, right? So they would actually have two copies of each gene on the X chromosome since they have two of them. So this could be an issue since there's two, you'd make double of all the proteins, but you don't want to make double of all the proteins because that can mess things up. And that can also cause you to waste important resources. So in women, in every cell, one of the two X chromosomes is going to be randomly disabled. So that way only one of them is functioning. So that is an interesting thing. So if you're interested in this, you can look up copy variants. So, or you could also look up gene duplication, because actually some genes can be duplicated. Like the DNA will actually the gene will actually double itself inside the DNA. It's pretty interesting, but not technically under gene expression. Okay, so back to our example. So cancer is when some of your cells start growing and dividing too fast. They eventually become a huge lump of bad cells that steal your nutrients and spread around your body, and eventually you will die unless you get treatment. So cancer often starts because of mutations in DNA, which lead to mutated proteins, which don't do their functions properly and end up doing something else. For instance, over 50% of cancers have DNA with a mutation in the nucleotides that makes p53 protein dysfunctional. Okay. So now I'm going to get into the topic of how we actually measure gene expression, because that is a really useful thing to know. Like, oh, I have a patient who has cancer, or I think they have cancer. What is going on in their body, right? Which genes are being overexpressed? Which genes are being underexpressed? For example, you'll often find that in people with cancer, genes that code for tumor suppressors, aka genes that make proteins that help prevent cancer, those genes are underexpressed. Less of those good proteins are being made. So one way that we can actually measure this stuff is using microarrays. So microarray is a square matrix. It's a chip. It's a very small chip. And there's thousands of these little wells or circles on the plate chip. And each of these little circles or wells contains a specific probe. And what a probe is, it's a complementary, it's a piece of DNA that is the opposite of another piece of DNA. So what this is meaning is that the common microarray has about 23,000 of these little circles. Let me turn on my annotation. The average microarray that's commonly used today has about 23,000 of these little circles. Inside each circle is a bunch of single-stranded DNA fragments. And remember that DNA is special because it can pair, it complementary pairs, right? So if you have a strand of DNA that says ATGC, this strand of single-stranded DNA, if you had this theoretically, it would pair and bind to another strand of DNA that's TACG. So the idea is that in each of these 20,000, 23,000 wells, there's a different DNA strand with a different sequence. This one might be ATGC, TGG, this one might be TTTGCA. And the idea is that each of these wells contains the DNA sequence that is the opposite of the RNA sequence of a gene. So there's 23,000 wells. That means you can capture the RNA products of about 23,000 genes. So for example, this one might be capturing the RNA for p53 protein. And this one might have the DNA that's the complementary strand for ABC protein. So it's a pretty clever method, taking advantage of the fact that DNA can bind to a strand that has the opposite nucleotides as it. So the application would be, let's say you have a healthy cell and you have a pathological or diseased cell. Let's say hypothetically the goal of the research is to find out differences in gene expression between lung cancer cells and healthy cells, because that would let us see kind of what are the differences in gene expression in these two cells, and how is that related possibly to the disease or treatment, or how can we help address these issues? Like, for example, you might want to see whether healthy cells have a higher level of tumor suppressor protein. So what you do is you take the cell, you culture it, and basically grow more of it. And then you're going to extract the RNA. I'm not going to go into procedure behind RNA extraction, but essentially you get out all the RNA from the cells. And then you're going to reverse transcribe it. So at this stage, before reverse transcription, you have a bunch of RNA. And the RNA is the intermediary between gene and protein. If you remember, it's the photocopied recipe. So what we're going to do is we're going to reverse transcribe it. And we're going to take a strand of RNA, and we're actually going to create the complementary DNA strand. And the process is called reverse transcription. This makes a lot of logical sense. So remember when we go from DNA to RNA, it's called transcription. So when you're going from RNA to DNA, it's reverse transcription. So now instead of having RNAs, we have a bunch of DNAs. And you're going to add a fluorescent little tag onto the end of each DNA strand. And I'll explain why this is important in a second. So now that you have a bunch of DNA, which is the copy of the RNA that is the complementary of a gene, you're going to mix them all together onto a microarray chip. You're just going to wash them over, like pour the DNA solution over. Next, you're going to use a laser and shine the laser through each one of the little wells. Good question. So reverse transcription is done not with ribosomes, but with reverse transcriptase, which is an enzyme. So ribosomes are not used in transcription. Ribosomes are used for translation. I can go back. So ribosomes take you from mRNA to protein. So ribosomes take in a strand of mRNA and build the corresponding protein. So that's what ribosomes are for. To go from DNA to mRNA, you use RNA polymerase. And there's a separate enzyme called reverse transcriptase, which allows you to feed in an RNA strand and build a corresponding DNA strand. I can get a picture of that. So reverse transcriptase is very cool. A lot of viruses actually use reverse transcriptase. I think someone won a Nobel Prize for discovering this. I'm not seeing a great picture. I mean, I guess this one is fine. Okay, so you have a strand of messenger RNA, the blue color, and the reverse transcriptase is going to chug along the blue strand of mRNA and add on the corresponding DNA nucleotide. So it's in red, so it's going to build the red strand, which is the opposite in terms of base pairs of the messenger RNA. And the difference is that it's DNA instead of RNA. So what you get at the end is a DNA strand called cDNA, and the C stands for complementary. The key thing to note here is to remember that this all works. Everything is working because adenine pairs with thymine and cytosine pairs with guanine. And using like one strand, you can always build like one, I don't know I forget the term, like a bijection. Like one strand of RNA or DNA has a single, it's like, there's like a 1-to-1 correspondence. Like if you have one strand of RNA, there's only one possible complementary strand of DNA that you can have. So knowing one allows you to know the other. I think there's a thing in math, maybe computer science, like bijection, something like that. Hopefully that makes sense. Okay, something else then. Okay, that math is not my thing. Okay, anyway, so we're shining a laser through the chip. And we added a fluorescent tag onto each of the DNA. So the standard method is to put green tags on your control group and red tags on your experimental group or disease group. And when you shine the laser through, if there's more of the red cDNA than the green tagged cDNA in a well, and remember each well is for like a given gene, if there's more red, then it's going to appear mostly red, right? If it's more green, that means that there was more of that cDNA present in the healthy cells. And if it's yellow, it means that it was about equally present in both cells. In some instances, commonly, you're not going to get any color because the gene is not expressed in either cell. So I forgot to mention this, but in the human body, all of our cells have the same DNA except for a few exceptions. And the way that we have so many different cell types like retinal cells, kidney cells, blood cells, whatever. It's because different genes are expressed in the DNA that gives each cell different characteristic. So there's going to be a lot of genes that just aren't expressed in either the cancer or the control, and those will just show up gray. And you get quantifiable numerical data from this by using a sensor that basically takes in the light intensity. So you're not just like looking at like, oh, red, red, green, green. This diagram is an oversimplification. So the reds are all the same shade, the greens are all the same shade, yellows are all the same shade. In reality, this wouldn't be likely. So this red might be slightly more red than this red. This yellow might be a little bit orangey, and this green might be less bright green. So it's like a gradient. Okay, next slide. So an example project you can do using microarray expression is to pick a disease and find an online data set from the gene expression omnibus and identify which genes are differentially expressed. So for example, in sick people, what gene is making more or less protein compared to a healthy person? And then what kind of biological functions are these genes involved in, and how could these genes affect the disease? So here's a paper I wrote as an example of what you can do. I'll just quickly open that up. So the goal in this example is to try to figure out what are some potential blood-based biomarkers or clues to detect Parkinson's. So just really brief overview. Parkinson's is a neurodegenerative disease, and it's usually by the time you can diagnose it, there's really obvious symptoms like you're shaking, you have slurred speech, and at that point, treatment is not going to help much. So it's useful to be able to detect if someone has Parkinson's earlier before there's all these obvious visual symptoms manifesting. So one idea is to try to find out if there's clues in their blood. So identifying messenger RNAs that are differently expressed in the blood of Parkinson's people versus control people. So the first task was to collect a variety of data sets from gene expression omnibus. I used 3 in this example. So in total, 94 Parkinson's disease samples and 45 control samples. Then you use a differential expression analysis tool called GO2R, which I'll demonstrate shortly. And it runs in your browser and it calculates the difference in gene expression between the control and Parkinson's groups and provides like a numerical statistic called log fold change. And log fold change is just a log base 2 of the difference in expression, the average difference in expression of experimental or AKA Parkinson samples over control samples. And finally, I did a network analysis, which is where you take the genes and you create an interaction map. For example, like, okay, this gene and this gene, their products interact. This gene creates an enzyme which modifies this gene's protein, and you can create a nice network that way. So yeah, so this is I did this one 2 years ago, so I was also learning myself, and you can get a lot more advanced with this stuff. So let's see. Is this what I want? Nope. Okay, here we are. So here's a more recent paper I did last year. And this one is about microRNA biomarkers for lung cancer. MicroRNAs are non-coding RNAs, so they don't make proteins. What microRNAs do is they find other RNAs and break them down or otherwise disable them, and they help reduce or regulate gene expression. So in this example, I compiled data from 4 data sets, with a total of about 4,000 samples. And I did some steps like a heat map showing the difference in expression across data sets of these microRNAs, a gene ontology. A gene ontology is essentially once you identify which genes are changed in your analysis, like, okay, these 500 genes have significantly different expression. You feed them to this tool called Panther, And Panther will tell you, oh, out of the 500 genes in your list, 400 of them were involved in cell division, right? So 400 out of your 500 genes were involved in cell division. Now, if we were to assume that you had a completely random list of genes, we would only expect to see about 25 genes involved in cell division. So the fact that your list of genes had such a high overrepresented amount of genes that are involved in the cell division process makes me think that whatever you're studying has a statistically significant effect on cell division. Personally, I think it's I'm a little bit skeptical sometimes about how reliable this kind of analysis is, but it is pretty useful just to see like, okay, a lot of my genes were involved in heterochromatin assembly, or a lot of my genes were involved in the microRNA metabolic process. And that makes sense, right? Because the idea of the research is to study microRNAs. Here's the gene network analysis I was talking about earlier, showing how the proteins of the genes interact with each other. And then here's a cool little graph about survival analysis. So these are called Kaplan-Meier plots, and they're really popular in the medical field. Essentially, they plot time against probability of survival. So as you can see, I had a few genes of interest, and I graphed the survival of people with low expression of the gene versus survival of people with high expression of the gene. So for example, in this graph, we see that people with low expression of the gene had greater survival probability than people with high levels of the gene being expressed. Remember, all these patients have only one copy of the IL-6 gene, but what is different is how many of the protein, IL-6 protein, is being made. Yeah, so that's basically what I'm going to get into now. Okay, and then so the final part of today's webinar is going to be a demonstration of how you can do the differential gene expression analysis. So the Gene Expression Omnibus, I've thrown that word a few times around so far. So the Gene Expression Omnibus is an online site hosted by the National Institute of Health, and researchers are encouraged to upload their data from microarray and sequencing experiments to the Gene Expression Omnibus for everyone to access. And I'm just going to use this one as an example. So it's Parkinson's disease versus controls whole blood. So you can click the Analyze with GeoTOR button. And it's a really, really neat tool. I think it's pretty well designed. You get this table of all the samples that are inside the data set. As you can see, some of them are healthy controls, some of them are Parkinson's disease, and there's even some that are neurological disease controls. So samples from people who don't have Parkinson's, but they might have Alzheimer's or Crohn's. Excuse me. So what we can do is we can select all the Parkinson's disease samples and assign them to a group called Parkinson's. So we got 50 of them. And then we can also pick all the healthy control samples and assign them to a group called controls. We got 21 of those. Oh, I missed one. Okay, 22 of those. So now we've told the online tool, we've told the computer which samples are my controls and which samples are my disease. Next, you're just going to click Analyze down below, and it's going to perform a differential expression analysis. So essentially what it's doing is for each of the samples that you have selected above in this selector window, it's going to find out the expression level it has for each gene, and it's going to perform a statistical test. So it's going to be like, okay, for this gene, ITCH, itch, you can kind of see that the Parkinson's samples look to have a little bit more expression than the control samples. and it's going to calculate a p-value, it's going to calculate an adjusted p-value, and a log FC. So this is what I mentioned earlier, the log fold change. It's the log base 2 of the ratio of expression between the Parkinson's group over the control group. Yeah, so I recommend using the adjusted p-value because it accounts for multiple hypothesis testing. So that is kind of an issue because there's like literally like 20,000 genes in this study. And if we're using like a 95% confidence cutoff for each one, like one out of 20 of those 20,000, we could be misidentifying. So that is a little bit more, you probably won't get that unless you've taken like a statistics course, and there's some younger students here, so I'm not going to go too much into that. But yeah, so essentially in a summary, what this tool does is it allows you to select 2 groups, Parkinson's, or literally any disease, cancer, COVID, influenza, whatever. And you can compare it against another group. And it's going to tell you, like, it's going to create these nice graphics for the genes. And it's going to tell you what is the p-value for, is there a statistically significant difference between the expression of your 2 groups. It's going to give you a log fold change value, which kind of gives you the ratio and the difference in gene expression as a number. And it tells you the gene and the full gene title. So if you want to play more around with this, like sorting by p-value, sorting by fold change, you can actually even download the full table, and it's going to give you the full 20,000. I can go over this one since we have time. So this is a volcano plot. A volcano plot is a cool little visual that plots the fold change against the p-value. The closer, each gene is a circle on this graph. So the closer a circle is to the center, 0.0, and the lower it is on the y-axis, the less significantly different it is. So like the very dense cluster of black circles, black dots, those are not statistically significantly different between the 2 groups. And that makes sense. The vast majority of genes are going to be very similar or no difference between Parkinson's and control group. Like for example, why would Parkinson's affect like toenail proteins, right? But the further away you get from the center of the x-axis and the further up you get on the y-axis, these genes are going to be a lot changed and a lot different between the 2 groups. So I went pretty fast with the demo, so you can actually get a step-by-step tutorial. So this Helix organization has created a COVID-19 bioinformatics step-by-step course. So it basically goes over what I just did, but step-by-step with explanations of each process. So it goes over how to find a data set of your own to look at. The example we use in this one is a COVID-19 infection data set where you'll compare gene expression changes in people with COVID-19 infection versus people who don't. And it just goes through all the button clicks, how to select data, etc. Now, if you prefer a video format rather than a text format, we also have a free gene expression course on udemy.com. It's pretty popular, good rating if I do say so myself. And in about an hour and a half, you'll learn how to do the gene expression analysis using GeO2R, and also learn how to use StringDB to make the network interaction maps and use Panther to do the gene ontology. And yeah, so these are both free. You can navigate to them by going to linktree/helix or helixscience.org. But yeah, so that concludes today's webinar. I hope you learned something, and thank you for coming. And let's just